But when ID card is not available, then the best method is third party identification. The person who is accompanying the examinee will give evidence for his identity. And you have to note down the detail of that person who is giving evidence for the examinee. Problem arises when these two things are not available. In case of certain mutilated bodies, unknown bodies are in living when the patient is unconscious. In that case, you have to gather the information, that is maximum information that is available from the examinee. And there are two methods. Number one is subjective method, and number two is objective method. In subjective method, subject means examinee is available and you can gather maximum information from that subject. Whether it is morphological data or data of his belongings. In morphological data, the anatomical data, physiological data, pathological data, all these things can help you to identify the patient. In anatomical data, the weight, height, or length in case of dead bodies should be noted. Genetic makeup, physical characters of examinee and his belongings, especially facial features, anything if you can find like glasses, cap, watch, anything should be noted in your record. Pathological parameters are any contracted disease, chronic disease like eczema, calcified fibroid, or scars of previous surgeries or injuries. In genetic parameters, blood groups, inherited morphological characters like Bohr bodies and Davidson bodies. Bohr body is condensation of nuclear material just along with nuclear membrane, and Davidson body is drumstick appearance of nuclear material inside the nucleus. Detail of these Bohr bodies and Davidson bodies will be given later. Then is the objective method. In objective method, again, understand this thing that in subjective method, you have the subject from which you can gather the information. Objective method is utilization of any morphological and belongings data. Say anything that you know that who is the tallest boy in the class. Every person in the class know about the tallest boy. This is only one physical character that is given to you, and you know the exact person. This type of information is analyzed by the agencies to isolate individuals' specific identity. And it is useful in decomposed bodies, mutilated remains based on collection of morphological and belongings data. And this belongings data may be biological or non-biological. Biological is any material that is derived from the living person, like hair, nail, bone. These exist and do not putrefy. And whenever you can get these things, wash them and make them fit for analysis in the lab. Non-biological material is clothes or other belongings. Clothes usually have any tailor mark or laundry box from where you can get information about the person. Trace evidence is any material which, when found on the body of the victim or assailant at the locus, that is scene of crime, or any other thing which acts as proof and helps in identification of source from where it has originated or derived and can be presented in the court is called trace evidence. To analyze this trace evidence is to establish personal identity or confirmation of provisionally identified person. Again, it can be biological or non-biological. Biological is originated from human beings or animal, mainly consist of body fluids or tissues like saliva, blood, semen, hair, nail, cerebrospinal fluid, and non-biological items are originating from the environment, like any fiber, grit, cigarette butt, paint, dust. There is principle known as low card exchange principle that gives you that every contact leaves a trace. And this contact results in exchange of the material from one place to the other. And this exchange is often two-way process. Comparing it with control will give you character of such a trace. For example, a case of zina. In trace evidence, you have to collect samples from the scene of crime. For preparation of these samples, there are two verbs, pre-lab work and lab work. Pre-lab work is more important, in which you have to collect the specimen from the scene of crime, reserve it, pack it, and transport to the lab. Then comes the lab work. For collection of specimen, collection should be proper and one must have the knowledge of the source of the specimen. And he must know what is to be collected. There are 4 phases when you visit at this scene. In first phase, just go through the history and documentation. Overview without touching anything at the scene of crime. Don't touch anything. Just take a bird's eye view and in the second stage, discover what has to be collected. Walk around twice around the scene and search for evidence to be collected and touch nothing. Make the area from where evidence is to be, mark the area from where evidence is to be collected. Just with imaginary lines, divide the area into different parts and mark every portion of that area. In third stage, you have to collect the material from marked area. Label that specimen and the with the marked area from where it is collected. After collection, when you complete the collection, confirm that no evidence is missed. Next is initially in old times. Another method was used for identification known as anthropometry or Bertillon system. Bertillon system. Bertillon was French assistant in criminal investigation department. He has devised it, and it depend on observations that no 14 different measurements from the body will be similar in 2 persons. So, अब सर मैं लेक्चर में हूँ, मैं आपको बताऊँ। And it was the first scientific method of criminal investigation that based on principle that after 21 years, most individuals' measurements remain same, like color of hair, eyes, complexion, shape of nose, ear, body measurements, height, AP diameter of head, span of outstretched arms, length of middle finger, index finger, sitting and standing heights, body mass like moles, scars, tattoos. And if you collect 14 such measurements, they will not match between two persons. But it was very hectic job for the collection of these measurements, time consuming. And it was replaced by dactylography, also known as Galton system. This was the surest method of personal identification that requires fingerprint experts for evaluation. Chinese used these thumbprints as ID marks on seals hundreds of years ago. In 1823, Czechoslovakian J. Perkinje described natural grooves on fingertips. In 1885, Henry Ford's Scottish working in Japan recognized the fingerprints and advocated the use of criminal investigation. Francis Galton worked properly on the subject and Edward Henry improved it. These fingerprint patterns are individual specific. 150 individual ridge characteristics on an average fingerprint that remains constant from birth to death. And these ridges are not described by desquamation of the surface epithelium or by abrasive use of sandpaper. And no two fingerprints have the same arrangements. These are formed by papillary ridges present on fingertips. These start appearing from 12th week of intrauterine life and completed by 24th week. There are four different patterns of fingerprints. Number one is arch. Number two, simple loop, double loop, or holes. There are four different basic patterns. Loops consist of about 60 to 70% of population, whereas bowl 30 to 35%, arch 5 to 10%, and composite 2 to 3%. In recording of fingerprint, there are two methods. Plain or rolled. In plain impression, just you have to touch the surface. That gives print of your fingerprints. Whereas in rolled impression, the whole finger is rolled from one edge to the other. In dead person, skin is made soft by oil or Vaseline. and it can be taken by injecting subcutaneous area with paraffin wax. Or boiling the fingertips in saline. FBI has 30 million fingerprints on its files, and none are alike. In our country, fingerprints obtained only at arrest of a person involved in crime or at the time of joining employment. Forensic experts at the time of autopsy sometimes requested to preserve whole hand or full finger or tips. If you have to preserve the fingertips, cut at proximal joints. Use separate containers for each and label them. In degloving injuries of skin, as in or in degloving in early decomposition, epithelium is spared from underlying tissue. Sparing the prints and preserved in between two glosses. This epithelium is taken and preserve these prints between two glass slides after air dried. Its medical importance is identification of criminals, identification of unknown and missing persons, identification for licensing procedures, and in immigrant cases. Identification data. Identification data is race, religion, sex, age, stature, teeth, fingerprints, footprints, lip prints, scars, tattoo marks, and personal clothing and contents of the pocket. Race and religion determined by clothing, eyes, hair, skull by measuring spelling index, that is maximum width between two masthead process into 100 over maximum length from supraorbital raised to occiput. There are different types of skulls: long-headed, medium-headed, and short-headed. Long-headed, also known as dolichocephalic, as in Negroes; mesocephalic, medium-headed, as in Europeans and Caucasians; and brachycephalic, short-headed, Mongolians. Primary parameters of identification: sex, age, and stature. In next lecture, we will talk about sex and age. Thank you.